Thank you for having me. I will talk about the logical next step for a Nextflow pipeline, which is once you're done coding, implementing it in a clinical setting. So I will first have a credit slide, which is also a presentation slide about who we are. Clinical Genomics Stockholm. We are specialized in translating research pipeline into a routine healthcare, but also developing pipelines ourselves. We are working in close collaboration with the Swedish healthcare system, especially the Karolinska University Hospital that is basically across the street from us. We are running about 4,000 to 5,000 genomes per year, and this is exponentially increasing. And we are seeing a lot more analysis getting into the routine clinical process. We are doing whole genome, whole exome targeted genome sequencing, metagenomics, microbial DNA, SARS-CoV-2 sequencing, and especially relevant for the examples that I'm going to talk about, the whole transcriptome sequencing. So I'm going to talk about one example with Annick Renevey and the clinical care of sarcoma. So a little bit earlier this year, a study was published about sarcoma, or patients suspected of sarcoma, and of a cohort of 200 patients that were offered whole transcriptome, whole genome combined analysis. It was found that in 7% of cases the diagnostic changed, which is quite significant, especially in soft tissue and bone tumors. That number went up to 15% of treatment-relevant variant identified. So that for sure was very concerning, and actions was taken with the press release of the Karolinska University Hospital that from now on routine clinical care will include whole genome and whole transcriptome combined sequencing. The part with whole transcriptome will be done with rnafusion, and this is a pipeline that I'm currently the main developer on. It is a very simple pipeline, a very simple metro map, and it has post-processing, very standard QC, and some visualization. I will not go any further into details. I will leave you with a bite-sized talk and the documentation as well as the Slack channel, but feel free to ask questions. And I will say that a release is soon to come with some major updates, I hope in the next few weeks. Now, my main point is how you go from a pipeline to an in-house medical device in compliance with IVDR. So when you have the pipeline, even your metro map, you're not finished. You have to also package it all together with pipeline dependencies, your software versions, your container versions, your visualization tools. What are you going to provide to the clinicians that they can actually pose a diagnostic? You have to build your documentation a little bit beyond what is the standard on NF Core. You have to version and to keep track of your references and database and your configurations, and in a similar extent as well, your resource management. And the goal of this book is to convince you that what is packaged in the pipeline is lighter to maintain, and that we can, as a community, build faster and better this type of clinically approved pipelines and medical device. There are a few important things to consider, which is traceability, maintainability, security, validation, documentation. I will mention a few things on each of those. So the first thing about traceability, we need to keep track of everything that was used. So of course there is versioning of software containers, reference sources, but not only this, also what you have done after with your reference that allowed you to run the pipeline, and your database, for example, if you if you have retrieved them at a specific time, you need to keep track of that so that you can reuse and make sure that you're always able to go back. So in the ideal case, what we want to do is have an isolated safe, put everything in there, running for as long as this is useful to us, then close it, put it in close the safe, put it in a shelf, and being able to open it back up, and everything works as it was intended, maybe in 5 years or something like that. So that we can make sure that we have everything documented and everything is accessible again. Now, there is the maintainability issue, and in my experience, URL changes and link breaks easily, and this is a problem that leads to a consequence that we have to do local storing of our references and databases in most cases. The security we are running on hospital servers, at least as the Stockholm healthcare region is not ready for the cloud. We are going more towards the data stays in the hospital. So there are some requirements that we have to obey in that respect as well, which is by a certain base image has to be used that lead to some conflicts, and there is going to be a routine security screening on the containers. And now there is a constant problem with really nice research-based software that is fantastic. All the clinicians want it, but it has not been updated in a few years, and it's not passing those security screenings. And then how can we risk mitigate? How can we update, make it easier for new developers to take over? So this is a constant theme. Yeah, I think that comes back. So about validation and assessments, so for each medical device release, we have to provide a validation report, and that summarize the results of a validation process. So we are testing if we're getting expected outputs, because you cannot provide say, okay, we are just providing you the results of this pipeline without actually testing that this pipeline does what it intends to do, and in which range can you expect it to work, with what kind of input you get what kind of results. So we are testing with the expected outputs with orthogonal methods. We are comparing also with previous analysis, and we are using for this commercial sample and also patient samples for whom for which consent has been given. And all of this is packaged into the reports that we submit to the accreditation unit, and hopefully that leads to an accreditation with a certain amount of revisions. Now we can also do benchmarking across medical device versions to see how the pipelines perform over time, and we can also open a few of these. So in the future, what we are going to tend towards is if we are going to optimize a few parameters for our data types and for our running process, we would like to make it open so that other institutions can see those and think if that might be something that they can also use, or they could test in a similar way. So we can share the validation procedure as much as possible, and that will alleviate the work immensely. We can share a certain number of meta information. Not everything is shareable, but it is we can find not loopholes, but ways around, workarounds. And there are, of course, comparison with commercially available samples. Now, of course, the validation is different if you want to use a research tool or a medical device. And how you combine those two different usage is always a balance. Now, we have also to consider documentation, which is operating instruction, the reports, the assessments. But in general, the community can build the documentation and provide benchmarking and the unit. The unit testing, you might test the pipeline in a different way that I would test it, and that actually does provide a lot of value. I will refer to you to the talk of Alexander Pelzer on Thursday about the work of the regulatory group. As a conclusion, I hope that I have presented to you our end accredited pipeline and what sort of requirements are needed in a clinical setting, what sort of things we can collaborate on and hope to speed up the process for other institutions. Of course, there is always this balance between research and clinical interests that will more and more, I think, be together in the same pipeline. So these two interests will actually cohabitate. And I hope you can see also the opportunities of collaboration for validation and documentation. With this, I will thank again the organizers and you all for your attention. Thank you.